So, without any further hesitation, I'd like to introduce our first speaker, Teresa Reyes. She's a research assistant professor here, and she's affiliated with both the neuroscience and pharmacology graduate groups. She got her BS, MS, and PhD all in the Department of Psychology at the University of Wisconsin in Madison. Her PhD in biological psychiatry focused on neuroimmunology, and during her postdoctoral training at the Salk Institute in La Jolla, California, she studied functional neuroanatomy. Her research interests focus on how prenatal and postnatal diet can impact development and brain functions, especially in the pathways that regulate appetite and reward. Her work has been featured in many top-tier journals, as well as in the popular media, including National Public Radio. Let's give a warm round of applause for Dr. Reyes.
Super. Thank you very much all for coming. I'm actually really thrilled to be participating in a Brain Awareness Week event. I've done 2 other events this week in my kids' schools. So earlier this week, I talked to a pre-K class about what the brain does in the morning. In the afternoon, I gave a couple of lectures to a series of 3rd grade classes. So we're moving up. We did pre-K, 3rd grade. So hopefully I've moved past that and I'm directing this at the right audience. Okay, so I'm just going to tell you 2 sort of short stories about some of the work that goes on in my lab. And this is really what I care about, which is how what we eat affects how our brain functions, brain development, and eventually behavior. Okay, so this is sort of the grounding of what I'm interested in. And this is a map showing the prevalence of obesity. And what you can see is that this is a statistic we're all familiar with, the prevalence rates of obesity starting in 1999, advancing through time to 2009, has rapidly increased. So there's a much greater percentage of the population that is overweight or obese. We're particularly interested in how this obesity epidemic affects particular time periods that are critical periods of brain development. So the first period that I'm going to talk about is how maternal obesity during pregnancy can affect offspring brain development. And the second period of time that is critical, sort of a critical brain development time period, is adolescence. So how does obesity during adolescence affect brain function and behavior? So let's first talk about the epidemic of maternal obesity. So in 2009, these are the numbers of women of childbearing age that were overweight or obese, so that you can see 29% of this population was overweight and 24% of this population was obese. So the majority of women of childbearing age were either overweight or obese. And when you actually look at the number of women who are obese or overweight who really were pregnant, that characterizes half of the women. So this is really something that affects the majority of babies who are born in this country. Along with obesity during pregnancy is a related condition, which is excessive gestational weight gain. So physicians have guidelines for how much weight is healthy for a woman to gain during pregnancy, and that's based on what their body weight was prior to pregnancy. We know that half of women who were normal weight prior to pregnancy gained too much weight during pregnancy, and 3/4 of women who had a high BMI prior to pregnancy gained too much weight during pregnancy. So these pregnancy conditions, obesity during pregnancy or gaining too much weight during pregnancy, again affect the majority of babies that are born in this country. So why is that a concern? Well, partly it's because babies who are born to moms who are obese or whose moms gain too much weight during pregnancy are at a much greater risk of being born large for gestational age, which is this LGA. So they're 3 times as likely to be LGA. And LGA babies are at an increased risk for obesity themselves. Those babies are more likely to develop more severe obesity, as well as learning and or behavioral problems later on. And so we were really interested in trying to model this in mice so that we could understand exactly how it is that the brain is changed in a way that might predispose these babies to the development of either obesity or behavioral or learning deficits later in life. So we use a mouse model where mice are fed a high-fat diet during pregnancy and lactation. So these mice are actually normal weight at the time of breeding, and they gain too much weight during pregnancy. So we're able to model this in mice. And just like in humans, these pups are born large for gestational age, and they maintain that heavier body weight throughout the period of lactation. And once they're weaned, their body weight eventually normalizes. Okay, so there are a lot of systems in the brain that control how we eat food. One of these is the sort of homeostatic system that regulates our food intake that is based on our metabolic needs, so eating when we are hungry. But there's this other system within the brain that sort of drives us to eat for reasons that are not related to our metabolic needs. So this is eating when it exceeds your metabolic needs. So eating because it's tasty, or because it's sweet or salty or sugary or fatty, or you're bored, or you're out for dinner, or you're tired, or all these reasons that we tend to eat that are not related to just being hungry. So the question was, and it's really this hedonic eating, which tends to be very calorie-dense types of food, that is really going to predispose for obesity or set up this obesity risk. So what we did was to look at the offspring once they were adults. So the moms ate a high-fat diet during pregnancy and lactation, and then we looked at the adult offspring. It's a very simple task in which we give the mice a choice to drink sucrose or water, or to eat a high-fat diet or a chow diet. All animals prefer either the sweetened solution or the high-fat diet, so that's a preference over 50%. But if the mom ate a high-fat diet during pregnancy and lactation, those offspring as adults have a significantly stronger preference for that sugary solution and that high-fat food. Okay, so then we wanted to look at some of the circuits within the brain to see if we could better understand how the brain is changed in a way that would underlie these behaviors. So we have this circuitry in the brain that tells us when something is rewarding, things like food, social interaction, for some of us exercise, for some of us when we're done exercising. And then this sort of system is what becomes dysregulated or co-opted by drugs of abuse. And there's just one example that I'm going to show you today, which is the opioids. So opioids are molecules that function within this reward circuitry. And when we looked at the expression of opioid molecules, so this is one of the ligands and this is a receptor, what you can see is that there was this significant dysregulation of this system in the offspring brain if the mom ate a high-fat diet during pregnancy and lactation. Okay, so let me just show you a short story about some of the other sort of neurobehavioral and cognitive learning endpoints that we've looked at. The reason we've done that is that there is data in the literature to support the idea that maternal obesity or pre-pregnancy BMI can also set up a risk in the offspring for cognitive or behavioral deficits. So to do this in the mice, we use a 9-hole mouse operant chamber. The mice, and it's amazing, they really do learn this task. They have sort of 5 holes that are activated, or that are present, and there's a light that will light up. And what the animal eventually learns is that when the light comes on in a specific hole, they need to go and nose poke in that hole, and then they will get rewarded with a little drop of chocolate yoo-hoo. And so I'm going to show you data from 2 tasks. One is called the progressive ratio, which is a measure of motivation, in which we can see how hard an animal is willing to work for that sweetened reward. And the second task is a test of executive function, which will let us look at things like attention and impulsivity. Okay, so here's what the animal has to do now in this task, is that they have to nose poke an increasing number of times to get the same single reward. So the first time they nose poke once, and they get their single drop of yoo-hoo, but the next time they have to nose poke 2 times, then 4 times, then 9 times. And so how hard an animal is willing to work is a measure of their motivation, because at some point an animal will say, I am not nose poking 27 times for my 1 drop of yoo-hoo. So what you can see here is that if the moms ate a high-fat diet during pregnancy and lactation, these offspring are significantly less motivated to work for the food, for the reward. And so remember, it's these animals that actually have a significantly stronger preference. So when they don't have to work for the sweetened food, they prefer it. So this is not just a function of them not finding it rewarding, but if they have to sort of act in a motivated way to acquire that behavior, to acquire that reward, they are less willing to do it. So they're kind of lazy. Let me just show you some data from the executive function tasks. So here, there are now 5 different holes. The animals have a choice across these 5 holes. And what they have to do is attend to the light coming on. And when the light comes on, they nose poke in that and they get their reward. So there are a lot of ways they can make an incorrect response. They can omit. The light comes on, they don't do anything. They can make an incorrect response to the hole that is not lit, or before the light even comes on, they can make a response. Omissions are a sign of inattention, while incorrect and premature responses are indications of impulsive-like behavior. So what we see in these LGA offspring is that they make significantly fewer correct responses, so they are not as good at doing the task. And when we look at what kind of mistakes they make, we see that there's no difference in their omitted responses. So it's not that they are inattentive. But if you look at their incorrects and prematures, these animals are making errors that look like impulsivity. So if the mom ate a high-fat diet during pregnancy and lactation, we see behaviors that look like impulsivity in those offspring when they're adults. We can also look at how quickly they learned the task. And what you can see, these are the LGAs here in this blue line, is that after about 12 days, all of the control animals had learned the task. But it took nearly 3 times as long. It took 30 days for all of the animals from the high-fat fed moms to learn the task. So in general, they were significantly slower to learn it. Okay, and lastly, let me just talk a little bit about adolescence. So now we are out of the prenatal vulnerability, and we're talking about consumption of a high-fat diet postnatally. The reason that this is important and that we care about it is that there's an increasing amount of data that shows that the chronic intake of highly palatable foods, high-fat diet, high-sugar diet, repeatedly is activating this reward system, and that repeated activation can change the way the brain responds to these rewarding stimuli. We've seen it in animals, and we've also seen it in humans. So if you take, this was done in college students, and you put them in, for example, a functional magnetic resonance imaging scanner, and look at how those people who are either obese or normal weight respond to a palatable milkshake. Subjects who are overweight or obese have a significantly blunted response to that milkshake. Okay, so the question that we were interested in asking was, we know that there are changes in this reward circuitry in response to a chronic high-fat diet. But is there sort of a critical period? So if that exposure happens during adolescence, is the adolescent brain more vulnerable? And are these changes reversible if you just take away the diet? This is the work of one of my graduate students who's defending her thesis in days. So she had these were her ideas, which were just great ideas. And this is essentially the experimental design, which is this, that animals were on a high-fat diet for 12 weeks, but it either started when they were 3 weeks of age, which would cover the period of adolescence, or it started when they were 6 weeks of age, which is more the point of young adulthood. They were all groups were on the diet for 12 weeks, and then we took them off the diet. Another group were taken off the diet for 4 weeks, and then there were animals who were age matched so that we had them at the correct ages. Okay, and I'm now going to introduce this concept of sex differences, which is that men and women are different. Okay, so here's what happens in a male brain. When these animals eat a high-fat diet over this 12-week period of time, and I'm just going to show you sort of snapshots. So this is gene expression within one area of the brain for all animals that started the diet at 3 weeks of age. So here you see this downregulation of these molecules related to reward in this area of the brain in male animals that have consumed the diet for 12 weeks. And interestingly, you see the exact same response in females. So in this way, males and females are not different. So then the question was, if we take the animals off the high-fat diet, can they recover? And actually, I was really surprised because they do recover, even though in 4 weeks their body weight has not recovered to normal control levels. So these animals are still obese. We've removed the high-fat diet, but now these changes in their brain circuitry have normalized. So that is really great news if you are a male, because that does not happen in the female brain. I don't know that it doesn't ever happen. It might be that the female brain is slower to recover and that maybe by 6 weeks or 8 weeks, we would see that recovery. But in this brain region at this time period, we didn't see recovery. So now when we look at the difference between 3 weeks and 6 weeks of age, again, the sort of genes that we looked at isn't particularly important, but you can see that there's this down regulation in response to the high-fat diet at 3 weeks of age. And when the diet was started as adults, you see this recovery when you removed the diet. But in this brain region and in these genes, if the diet started as an adolescent, and now these are males, the diet started when the animals were adolescents, this brain region was not able to recover. So in this way, the adolescent brain does appear to be more vulnerable to exposure to the high-fat diet. Okay. This is my last slide. This is what I care about, nutrition and brain development. And so I hope that what I was able to convey to you is that what the mom eats during pregnancy is really important, not only for her own health, but for the health of her baby, and particularly for the brain development of that baby. And what we eat throughout adolescence and even as adults can affect the way our brain works and functions and behaves. And so it's important. These are the people who did the work and the people who pay me money to do the work. So thank you very much.
We have about 5 minutes for questions, and we have microphones on either side if you guys want to chime in.
So you were very meticulous in your talk about mentioning that the moms were fed a high-fat diet during gestation and lactation period. Has there been any further study to see like if only gestation or only lactation period can affect the brain development as well?
Yeah, it's a great question. And the reason that we do gestation through lactation in rodents is that we want to try to more fully match what happens in a human pregnancy and brain development in the rodent. So rodents are born with a more immature brain so that the first 3 weeks of life is more akin to the third trimester of a human. So that was why we did that. But in these rodent studies, there have been other groups who've split out the lactational period versus just the gestational period. And it's very clear that what happens in lactation also is important in its own right. We've also done work to look at whether or not obesity prior to pregnancy, in the absence of obesity during pregnancy, can have any of these adverse effects on the brain, and it does. So that risk can be imparted even prior to pregnancy.
You mentioned interesting sex differences in your adolescent studies, and I was wondering if you found any of those sex differences in your prenatal studies.
We have. So really, just about anything you do in prenatal work, you will get an interesting sex difference. The behavioral work, though, the attentional, the impulsivity that we've seen, we see that in our female offspring as well. And we see, so actually in those endpoints, they actually are very similar, as well as the preference data. Although in the females, we see a much stronger preference to sucrose and not so much the fat preference, whereas we see both fat and sucrose in the males. So there are some differences. And we see more sex differences in some of the endpoints that we look at that relate to obesity, so metabolic rate and sort of basal food intake and activity, those sorts of things.
Thank you.
Hi, thanks. I was wondering if you've taken this out to further generations, the prenatal stuff. Have you looked into the grandchildren of these mice and seen if the effect lasts if you take the children and keep them on a regular diet?
Yeah, exactly. Super interesting area of research. We have not done it yet. We think about it. We actually thought about one study to do with, we had just a cohort of very obese males that still existed in the lab. So we said, hey, let's just see, if we can look at male, transmission of this phenotype. And I think we bred 8 obese males and 8 normal weight males, and none of the obese males sired pregnancies. So it was like, okay. They were very old though too, so that was so old. But that in itself was sort of like, well, that's interesting. We should probably follow that up. But so it's something that we think about. But there are other groups that are doing that.
I think we have time for one more question.
Thank you for your talk, Teresa. My question was, what are your thoughts on placental reprogramming as a possible link between what mom eats actually is important for the brain development of the pup?
Yeah, the placenta is a really fascinating organ, obviously, that develops only during pregnancy, but it's, the point of communication between what's in the mom and how that gets translated to the baby. And so there is a lot of really interesting work showing how the placenta changes in response to maternal high-fat diet. And there are very clear changes that involve oxidative stress, and there's even morphological changes. And so I think that's an important conduit that could be transmitting this phenotype. But I do think that there's also probably specific components like fatty acids in the diet that are actually getting to the baby, so that it's not just mediated by the placenta, but placental dysfunction is part of the phenotype. Okay, thanks.